Important clinical SAS interview questions are: 1. Identifying the baseline value and baseline flag. 2. What is post-baseline value? That is POBL. 3. Calculate change from baseline. That is CHG. 4. Calculate percentage change from baseline. That is PCHG. 5. Calculate last observation carry forward. That is LOCF. We will check all one by one programmatically. Baseline identification. Last non-missing value before the treatment start date, or the closest value to the treatment initiation. Before any treatment is administered, we measure key health parameters like systolic and diastolic blood pressure. These initial readings are not just numbers. They represent the state of a patient's health prior to the intervention. In trials of chronic conditions, baseline measurements are vital for determining whether a treatment can alleviate pre-existing levels of pain, anxiety, hypertension, and similar conditions. The baseline serves as a control point for future comparisons. Post-treatment. Following the treatment phase, these health parameters are measured again. The post-treatment values are then compared to the baseline to determine the treatment's impact. Check the clinical significance here. We will discuss about 1. Baseline identification. 2. Calculate change from baseline. 3. Calculate percentage change from baseline. Under baseline identification, first 1. Identify the treatment start date. Sort the records. Manually flag baseline records. And finally create a baseline flag. Second 1 is calculate change from baseline. Change from baseline equal to post-baseline minus baseline value. For example, if the baseline is 10 and the post-baseline value is 15, the change is 5. Third 1 is calculate percentage change from baseline, which is percentage change from baseline equal to change from baseline divided by baseline multiplied with 100. For example, if the change is 5 and the baseline is 10, the percentage change is 50%. We will see first question, identifying the baseline value and baseline flag. A BLFL means analysis baseline flag. Baseline value is the latest record with non-missing result, on or before treatment start date for each parameter within a subject. The baseline flag serves to mark the initial status or condition of each subject at the beginning of the study. It captures baseline measurements of key variables such as demographics, medical history, disease severity, vital signs, laboratory values, and other relevant parameters. Here baseline means the drug given date, or before that date. Here in diagram drug given date is 9th January. Cholesterol measurement has not taken on the same date, so prior date 7th January is taken as the baseline date for baseline value. Please check the below diagram for the creation of analysis baseline flag condition. Baseline flag is equal to yes when analysis date is less than or equal to treatment start date. Post-baseline flag equal to yes when analysis date is greater than treatment start date. Check below record for the analysis baseline flag derivation. Baseline value is the latest record with non-missing result, on or before treatment start date for each parameter within a subject. Here sort the lab date with descending order, and pick the highest date record. Then using if first clause, put baseline flag to 0 next using if condition when analysis date, that is lab date is less than or equal to treatment start date and analysis value, not equal to null value put the baseline flag to yes. We will check second example for the baseline value. Here first separate the dataset as baseline and post-baseline datasets. When visit n is less than or equal to 2 output all records to the baseline, and other records in post-baseline dataset. In baseline dataset analysis value HR should not null value. Finally merge baseline dataset with post-baseline, and finally derive change from baseline. Now we will see creation of baseline value, post-baseline value and calculation of change from baseline, percentage change from baseline. Here are blood pressure data set. Sort both data sets and merge it. Sort the data by subject, test, and week. Next calculate change from baseline of systolic blood pressure and diastolic blood pressure values. Next carry forward baseline results and finally determine baseline and calculate changes. We will see now how to calculate last observation. Carry forward that is LOCF, which is very important to the clinical SAS interview. Last observation carried forward, LOCF is a statistical method for analyzing data with missing values in longitudinal studies. How it works. Last observed value is used to replace missing values. The imputed and observed data are then analyzed. Advantages and disadvantages. LOCF is considered conservative, but it can introduce bias in certain circumstances. Some say that LOCF is not valid under general assumptions, and is not based on statistical principles. LOCF has been used in many studies, including randomized controlled trials. However, some say that the published results of some randomized controlled trials of dementia drugs may be inaccurate or invalid due to bias introduced through the use of LOCF. We will see two examples for last observation carried forward. We will check first example. Weight is a SAS data set. Variable analysis value is having missing values, which are imputed programmatically. First filter the data set where visit is less than or equal to 3. Retain the last observation carried forward variable to null value. Then calculate LOCF equal to weight if weight is not equal to null value. Check second example for last observation carried forward. Here one input is cholesterol data set and the other input is pill dosing data. Cholesterol data set variables are patient number, lab sample date, HDL, LDL, and triglycerides. Dosing data set variables are dosing date. Sort both data sets and merge them. Next define baseline, HDL, LDL, and triglycerides variables. Next set up arrays for baseline variables and lab values. Next initialize baseline variables to missing. Finally, if lab value are within 5 days of dosing, retain it as a valid baseline value, and keep last record per patient as these hold the LOCF value. For complete training details, please contact us and visit our website crystalclinicalsas.com and mail us to clinicalsas924@gmail.com. Enjoy your training at our institute and explore more knowledge. Thank you. Bye-bye.